Hi, my name is Daniel. I'm working at Intiquan, a company located in Basel, Switzerland, offering high-quality integrated pharmacometric consulting services. QSP models are increasingly used in the pharmaceutical industry because they allow to mathematically represent more of the biological background knowledge about the disease, the cellular processes, and the mechanism of action. Accordingly, one is hoping that these models are better suited for extrapolation, for example, from animal to human, or from one drug to another. However, and here we're getting to the big picture, ultimately one needs to extend the mechanism-centered QSP models to a full-fledged population model connecting the mechanistic and the clinical levels. This step bears a certain challenge, both in terms of concepts and in terms of software. Let us have a look at the current state of QSP modeling in the pharmaceutical industry and compare this with our in-house situation at Intiquan. On the one hand, tools like Berkeley Madonna and SimBiology are used. These software packages are great for simulation of hundreds of differential equations with hundreds and thousands of parameters, but they are not dedicated to parameter estimation. These tools are dedicated to handle the complexity in the model. When it comes to parameter estimation, companies use non-mem or monolix. The software successfully handles hundreds and thousands of subjects, but has quite some limitations on the size of the model and number of parameters to be estimated. Clearly, there's a disconnect between the mechanistic and the population level applications. We at Intiquan have developed a QSP modeling module to close this gap. I say module because it is integrated within our modeling and simulation framework called IQR tools. IQR tools simply connects to other software like Berkeley Madonna or SimBiology, allowing you to import models from there. It even lets you reconnect to non-mem or monolix and lets you run your estimation there if needed and if possible. Let's have a closer look at our QSP module. The modeling syntax we use is very similar to the one used by SimBiology or Berkeley Madonna. SBML import is supported. For robust parameter estimation, we employ symbolically derived sensitivity equations. These allow to use gradient-based optimization methods, converging to the optimum more reliably, more reproducibly, and in less time. We combine local optimization with the global search algorithm that explores the space of possible model realizations, allowing us to deal with model uncertainty. Especially for larger models, parameter uncertainty and identifiability is a big topic. We have implemented a fast and efficient integration-based approach for likelihood profiling. This approach allows to spot non-identifiable parameters while providing structural insight. Finally, variability in the parameters to represent virtual populations is implemented via a penalized maximum likelihood approach. To illustrate the capabilities of our software, we have worked through a published model with 8 ODEs and with published data of 18 subjects. The model was originally coded in SimBiology and was imported to IQR tools. To identify the most relevant parameters, we used the IQR tools functionality to perform a sensitivity analysis. Due to the limited availability of data, we did not expect that all model parameters could be estimated. To identify those parameters most crucial to adequately describe the data, we used the IQR tools functionality to conduct a stepwise parameter modeling approach, SPM, in analogy to the stepwise covariate modeling approach, SCM. Based on the iterative approach, we were able to identify the 6 most crucial parameters. The plots in the box show individual fits. Random effects on all estimated parameters were considered. Goodness of fit plots are shown. In the setting of 6 parameters, monolix was applicable, and we decided to check our estimation results with those generated by monolix. We found almost identical results, both in terms of population parameters and simulations based on variability. In conclusion, we successfully fitted a typical mechanistic QSP model to individual level clinical data, adequately described the variability in the population, and reused our calibrated model directly for the simulation of new drug regimens for a virtual population. The example chosen for this presentation was relatively small, with only 8 ODEs and 18 subjects. In our current projects, we successfully apply our methods to ODE systems with around about 100 ODEs and up to 400 subjects. We hope that the smaller example was illustrative and incentive for everybody to cross the border, make your mechanistic QSP model ready to deal with clinical data and virtual populations. We are happy to help and support you with this step. If you are interested, please don't hesitate to get in touch with Intiquan.